Hello everyone, I am Shaileyee welcoming you to the Medical and Pharmacy Crash Course Division in the Knowledge Club online. Friends, today I have come with an episode in the Medical and Pharmacy Crash Course Division, the quick revision of Cell Structure and Functions chapter. This questions and answer session will help you to ready for any kind of competitive exam or medical exams. I divided quick revision of Cell Structure and Functions chapter in 3 parts for making the short episodes. This is the part 1 episode. Definitely, these episodes help you to revise, consolidate and polish your knowledge and skills. Also help you to develop an in-depth understanding of the subject, assess and review knowledge. Suggestions contained in these type of episodes have been designed to be similar in the pattern used in the JEE Medical, NEET, CSIR NET, GRE, DBT, JRF, ICMR, GATE, GPAT, JNU PhD, JU PhD, IAS and so on medical exams and pharmacy semester exams. For the sake of better self-assessment as well as a thorough and comprehensive understanding of the subject, I have kept it topic and chapter wise. Stay tuned up to end because if you skip the lecture, you can miss a valuable points in between lecture for your exam. So stay tuned up to end. I am going to start today's episode. Let's start. Point 1. Active transport. Form of transport across the cell membrane that requires input of cellular energy. Point number 2. Amphipathic. Describes a molecule that exhibits a difference in polarity between its two ends, resulting in a difference in water solubility. Third point. Anaphase. Third stage of mitosis and meiosis, during which sister chromatids separate into two new nuclear regions of a dividing cell. Fourth point, anticodon. Consecutive sequence of 3 nucleotides on a tRNA molecule that is complementary to a specific codon on an mRNA molecule. 5. Autolysis. Breakdown of cells by their own enzymatic action. Point 6. Autophagy. Lysosomal breakdown of a cell's own components. Point 7. Cell cycle. Life cycle of a single cell from its birth until its division into 2 new daughter cells. Point 8. Cell membrane. Membrane surrounding all animal cells composed of a lipid bilayer interspreaded with various molecules also known as plasma membrane. Point 9. Centriole. Small, self-replicating organelle that provides the origin for microtubule growth and most DNA during cell division. 10. Centromere. Region of attachment for 2 sister chromatids. Point number 11. Centrosome. Cellular structure that organizes microtubules during cell division. Point number 12. Channel protein. Membrane spanning protein that has an inner pore which allows the passage of one or more substances. Point number 13. Checkpoint. Process point in the cell cycle during which certain conditions must be met in order for the cell to proceed to a subsequent phase. Point number 14. Chromatin. Substance consisting of DNA and associated proteins. Point number 15. What is chromosome? The condensed version of chromatin. Point number 16. What is cilia? Small appendage on certain cells formed by microtubules and modified for movement of materials across the cellular surface. Point number 17. Cleavage furrow. Contractile ring that forms around a cell during cytokinesis that pinches the cell into 2 halves. Point number 18. Codon. Consecutive sequence of 3 nucleotides on an mRNA molecule that corresponds to a specific amino acid. Point number 19. Concentration gradient. Difference in the concentration of a substance between two regions. Point number 20. Cyclin dependent kinase CDK. One of a group of enzymes associated with cyclins that help them perform their functions. Point number 21. Cyclin. What is cyclin? One of a group of proteins that function in the progression of the cell cycle. Point number 22. Cytokinesis. Final stage in cell division where the cytoplasm divides to form two separate daughter cells. Point number 23. Cytoplasm. Internal material between the cell membrane and nucleus of a cell, mainly consisting of a water-based fluid called cytosol, within which are all the other organelles and cellular solute and suspended materials. Point number 24. Cytoskeleton. What is cytoskeleton? Skeleton of a cell formed by rod-like proteins that support the cell's shape and provide, among other functions, locomotive abilities. Point number 25. Cytosol. Clear semi-fluid medium of the cytoplasm made up mostly of water. 26. DNA polymerase. Enzyme that functions in adding new nucleotides to a growing strand of DNA during DNA replication. Point number 27. DNA replication. Process of duplicating a molecule of DNA. Point number 28. Diffusion. Movement of a substance from an area of higher concentration to one of lower concentration. Point number 29. Diploid. Condition marked by the presence of a double complement of genetic materials, the two sets of chromosomes, one set inherited from each of two parents. Point number 30. Electrical gradient. What is electrical gradient? Difference in the electrical charge, the potential between two regions. Point number 31. Endocytosis. Import of material into the cell by formation of a membrane-bound vesicle. Point number 32. Endoplasmic reticulum. Cellular organelles that consist of interconnected membrane-bound tubules which may or may not be associated with ribosomes, rough type or smooth type, respectively. 33. Exocytosis. Export of a substance out of a cell by formation of a membrane-bound vesicle. 34. Axon. One of the coding regions of an mRNA molecule that remain after splicing. Point number 35. Extracellular fluid (ECF). Fluid exterior to cells includes the interstitial fluid, blood plasma, and fluid found in other reservoirs in the body. Point number 36. Facilitated diffusion. The diffusion of a substance with the aid of a membrane protein. Point number 37. Flagellum. Appendage on certain cells formed by microtubules and modified for movement. Point number 38. What is Z0 phase? Phase of the cell cycle usually entered from the G1 phase, characterized by long or permanent periods where the cell does not move forward into the DNA synthesis phase. 39. G1 phase. First phase of the cell cycle after a new cell is born. G2 phase. Third phase of the cell cycle after the DNA synthesis phase. So guys, hope you enjoyed today's important term session and increased your knowledge more on cell structure and functions. I am coming with another division session on this chapter part 2 in next episode. That also will be help you to flourish your knowledge more. If you like this video, please like, share and comment freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. After subscribing our channel, please click on the bell button for getting the regular updates. I am coming with another video very shortly. Till then, goodbye.